Right. Hello, and welcome to the very first seminar series of the Women's Health, the Research Cluster for 2025-2026. It's my pleasure to introduce to you Dr. Holly Hunsberger. We'll be talking about sex analytics and psychosocial stress, vulnerabilities to aging and Alzheimer's disease. Before we do so, I wanted to do a land acknowledgement. As soon as I can advance my screen, there we go. We recognize that we live, work, play, and participate in a community on the unceded, ceded, and traditional territories of the 630 First Nations, each of which possess their own unique traditions and history here on this land that we now refer to as Canada. And I recognize that not all of you are coming or live in Canada. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action and the United Nations Declaration on the Rights of Indigenous People. In all of our work, we're committed to ensuring Indigenous women's rights to health and safety and the equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Today, I'm joining you from Toronto, Ontario, Canada, which is covered by Toronto Purchase number 13 with the Mississaugas at the credit. Toronto is now home to a vast diversity of First Nations, Inuit, and Métis who enrich the city. Many of our members also live in Vancouver, which is part of the unceded homelands of the Coast Salish peoples, the Musqueam, Squamish, and Tsleil-Waututh First Nations. So this is brought to you by the Women's Health Research Cluster. Who are we? We have over 950 members across 40 countries. We run a number of events. There'll be a trainee research presentation series, I believe, on hormonal contraceptives, probably sometime in October. So please keep a lookout for that. We are on all the social media channels that you could possibly want to be on. And we give out a number of trainee travel awards every year. There's two competitions a year, so please apply for that. If you're presenting your research at a conference and need a little bit of help, and become a member if you're not already a member. And with that, I'm going to stop sharing my screen so that Dr. Huntsberger can start sharing her screen. And while she does that, I'm going to read out her bio, and I can't wait to tell you what her fun fact is. So, Dr. Hunsberger obtained her PhD in behavioral neuroscience from West Virginia University under the mentorship of Dr. Miranda Reed, where she studied glutamate's role in Alzheimer's disease. From there, she secured a postdoctoral T32 fellowship from Columbia University with Dr. Christine Denny, where she discovered her passion for understanding women's health. Yay! And we're very thankful for that throughout aging and Alzheimer's disease. Her initial postdoctoral work examining sex differences in anxiety and cognitive decline in mice with Alzheimer's disease mutations earned her a K99 ROO Career Transition Award through the NIA, which is the National Institute for Aging. For those of you in the US, you'll be very familiar with those terms, but if you're not familiar, please just put a question in the chat. This translational work was recently published in Biological Psychiatry. Holly began her faculty position at the Rosalind Franklin University in November 2021. In her laboratory now, Holly continues to examine sex differences in Alzheimer's disease, as well as the Alzheimer's disease exposome, which encompasses the unique experiences of each individual, including their genetics, lifestyle, injuries, education, medications, social networks, and socioeconomic status. And each of these plays a role, but it's unclear how, and this is what her lab will uncover. Now, her fun fact, I told you you're going to be amazed, it's pretty amazing. She's saying, God bless America for the Philadelphia Phillies National League baseball game. Now, not many of us can claim to have done that. When was that, Dr. Hunsberger?
The year, I think that was around 2014.
Wow.
So a while ago now, but
Watch out, Taylor Swift.
yeah, I don't know about that, but yeah, I was a singer in a past life, so
Great. Well, maybe we'll get a little bit of a sing-song, who knows? With that, please begin whenever you're ready, Holly. Thank
great.
you.
Thank you. Thank you for the introduction. Let me minimize Zoom here. Okay, perfect. Oh, hmm, okay. Yeah, so thank you, Lisa, for the introduction. Thank you to the cluster for inviting me. I'm really honored to be here today and to talk about what my lab has been up to for the past almost 4 years now. And I'm going to touch upon a lot of different topics today, all within what I call the Alzheimer's exposome, and I'll get into what that means. But really, just looking at how all of these can contribute to vulnerabilities in aging and Alzheimer's disease, everything through a sex difference lens. And so, before I get into the science, I just want to say that I'm also really proud to have started my lab at RFU. It's the only U.S. health science university named for a woman, and it's been such a great experience being here. So the Alzheimer's exposome is really taking into account individual differences throughout aging. So the reason that Alzheimer's is so hard to study and so hard to cure is because people go through life with so many individualized experiences. And it's really important to think about Alzheimer's disease as a multifaceted disease and not really focusing on one specific area, but looking at how all of these factors can contribute to disease progression. So you can think of things like exogenous factors, so your environment, how you grow up, endogenous factors like genetics, your metabolome, and behavioral factors such as your lifestyle and stressors. And so our lab really focuses on these behavioral factors with a little bit, oh, sometimes it's letting me, yep, there we go. With a little bit of endogenous, looking at those sex differences there. So my talk is going to go through these topics on the left. So we're going to talk about sex differences in Alzheimer's disease progression. We're going to talk about how anxiety can influence this. And then we're going to talk about drugs that treat this anxiety and whether they are beneficial or detrimental. And then lastly, I'll talk a little bit about psychosocial stressors on top of all of these things. And so I don't have to convince this crowd, but of the millions of people suffering from Alzheimer's, 2/3 are women. That's not just because women live longer, it's true across the lifespan. And more importantly, it's the disease progression that really matters. So yes, both men and women will succumb to the same cognitive decline, but how they get there is vastly different. And also the pathology can differ throughout the trajectory. So we really need special personalized therapeutics and also could be helpful for diagnostics as well to better understand these transition in both men and women. And so just to get everyone on the same page of Alzheimer's disease, this is a neurodegenerative disorder, results in cognitive decline. And at the end stage, biologically, we see amyloid plaque accumulation, intracellular tau tangle accumulation, and these together contribute to neuronal loss. But we're really interested in the behavioral changes that are occurring many years before this downstream pathology because we're really interested in early interventions. And so we've decided to look at a lot of the behavioral changes. It turns out that these behavioral changes are not just comorbid with Alzheimer's disease, but actually can occur several years before Alzheimer's pathology, giving us a little bit of a window to find therapeutic targets and better understand the pathology and trajectory. And so we became really interested in this idea of neuropsychiatric symptoms such as anxiety, depression, apathy, sleep disturbance, and really honing in on anxiety because we also know that anxiety disorders are more prevalent in women. And so when I first set out to answer this question, I really wanted to understand how do biological sex and these neuropsychiatric symptoms drive Alzheimer's pathology and progression? And also, can we use these symptoms as predictors of disease or even as treatment targets? And so all of the studies that I'm going to talk about today, we're using the APP-PS1 mouse model of Alzheimer's disease. So this is an amyloid-only mouse model. It's an overexpression model. So I do like to note that limitation. But it does show progression similar to humans with plaque pathology buildup, neuronal loss, and cognitive impairment. It's just on an accelerated timeline. And so I won't go into too much detail, but really just wanted to set the foundation for my talk on where I'm going in my lab now. But if you are really interested in the details, feel free to scan the QR code there because this study was recently published. But we were looking to identify sex differences, in Alzheimer's disease in an AD mouse line. And the reason I even got into this area of work is because in graduate school, we were looking at female mice only. And then in my postdoc, we were looking at males only. And I thought, well, why aren't we looking at both? And so this was about 2016, right, when sex as a biological variable was included at the NIH. And so it was perfect timing for me to go into sex difference research. And if you notice on the slide before this, the cognitive decline in this model was pretty late. It was about 12 months. It turns out that was all done in male mice. And so when I was running these experiments, I thought I was completely wrong because I was seeing such early cognitive decline. But it turns out they just hadn't looked at female mice. And I also want to note that these mice are on a 129 background compared to a C57, which is what you would typically buy from JAX. So that could make them a bit more susceptible to neuropsychiatric symptoms and cognitive decline, which makes it actually a very perfect model for the questions that we're asking. And so what we found was earlier anxiety in this mouse model in the AD females. We did see anxiety with age in males. So it's not that they don't have anxiety, it's just that the progression differs. It comes on later. And we also saw earlier cognitive decline in the AD females around 2 months of age, which was similar time with different anxiety tasks. And then we also saw that cognitive decline in the males even earlier, around 4 to 6 months of age, compared to what had been published previously. And so we looked at a lot of different types of anxiety here: avoidance, perseverative, hyponeophagic behaviors. And then for memory, what we looked at was contextual fear conditioning. So training the mice with shocks and then re-exposing them to the same environment without shocks to measure freezing behavior, which we use as a proxy for memory. And you'll see a lot of these behavioral tasks throughout the talk as well. So from these behavioral studies, we wanted to know what was happening in the brain. So unfortunately, we didn't take brain tissue at 2, 4, 6 months of age when we first did this. We ran behavior at 2, 4, 6 months of age. We only took brain tissue at the end at 6 months of age. And so what I'm showing you will be kind of a later time point, and we're missing those early time points, which is some of the areas that we're heading in next. And so to look at changes in the brain, what we did was we crossed our Alzheimer's line with an activity-dependent tagging line, the ARC-Cree mice. So this mouse line was developed by my postdoc mentor, Denny. Essentially, it allows us to tag memory engrams across the entire brain. So this is a genetic model. It's an indelible label. It allows us to tag cells that are active during the encoding of a memory in green. We can then, like I said, re-expose those animals to a similar context, take brain tissue 1 hour later, use the power of immediate early genes such as CFOS here. Oh, it keeps wanting to advance there. Okay. And we can use CFOS, which is an immediate early gene here, to look at cells that come online right after an experience. And when we look at the final product, any cells that were important for encoding and retrieval of that memory are termed a memory trace or a memory engram. And so we decided to look one at all of these cells across the entire brain, and then also at memory engrams specifically in the hippocampus, which this is what I'm showing you right here. So this is a rodent hippocampus, dentate gyrus, CA3, and CA1. And so what we found were these sex-specific brain-wide network changes throughout AD. I hope you can appreciate that even at baseline, control male and female mice are different here. This is after a fear conditioning paradigm. So this would be the memory retrieval. These are brain-wide networks. So we're looking at the correlation of CFOS, that immediate early gene, between all of the brain regions. The blue and pink are positive correlations, the gray are negative correlations, and the thicker the line, the stronger the correlation. And so in AD females, we saw a very disconnected brain network at 6 months. At males, compared to their controls, we actually see kind of a hyperexcited brain-wide network. And so this has been seen a lot early on in many Alzheimer's patients. And what I think is going on here is that because the AD female mice seem to be on an accelerated progression timeline, they perhaps show this excitability earlier than 6 months, and we're capturing a little bit of a late time point here. So then we delve a little deeper. We look at the hippocampus, and this is where we counted our memory engrams, and we also saw sex-specific differences here. So we see a decrease specifically in the dorsal CA3 of those engram cells in female AD mice. We see the difference in the DG in male AD mice. And so with all of this, we really wanted to make sure that what we were seeing was translatable to humans. So our lab is very interested in translational science. And because we do use an overexpression amyloid-only model, we want to make sure that what we're seeing is true. And so we turned to a biostatistician, Dr. Sanjo Lee. We use the Alzheimer's Disease Neuroimaging Initiative data set to understand anxiety in male and female subjects. And so we saw similar to what we saw in mice, which was really exciting. We saw an increase in anxiety, typically in AD patients. That was true for both males and females. But what was interesting is that patients that were positive for anxiety and also positive for amyloid in their cerebral spinal fluid showed a faster transition here in these dotted lines to dementia. And it was significantly faster, specifically for women. We then looked at age, brain atrophy, and anxiety, combining males and females, to see what was the strongest driver of dementia transition in this cohort. And again, we saw anxiety as the number one predictor, which was very interesting and exciting. So again, I just want to note this is a limited study. ADNI, at the time when we had analyzed this, primarily white males, highly educated. And so we really want to see if this will hold true for other types of large data sets. And we are starting to see people publish on this and seeing similar patterns. So now I want to kind of go into what my lab has been doing. We saw these anxiety-like behaviors we saw earlier in the females, but we don't really know what's going on in the brain earlier. For males, we see a lot of these behavior changes later at about 6 months, but maybe we're missing something earlier that could be changed in males. We didn't really look at depressive-like symptoms. And again, we also don't know what's going on at that earlier time point. And so I have this hypothesis that these emotion and cognition circuits are kind of driving these behaviors and these pathologies because they work hand in hand. And we became very interested in the ventral CA1 region because we have some preliminary data to show that those memory engrams, the more activated they are in the ventral CA1, the worse these Alzheimer's female mice perform in that fear conditioning task. And so that's a little bit opposite of what you would think, but if you think of some of the hyperexcitability hypothesis early on in Alzheimer's disease, it doesn't, just because something is working harder doesn't mean it's better. So we saw a lot of changes in the ventral CA1 here that we didn't see in any of the other groups. And then we also see a lot of FOS activation in the ventral CA1 in the AD female mice as well. So we've become very interested in this region. We're also interested in the connections to the amygdala because very important for anxiety-like behaviors as well as cognition. We think there's something to this circuit that's happening in Alzheimer's disease, specifically in females. And we're also interested in inhibitory circuits. So from here, we are looking at the connections between the ventral hippocampus, amygdala. We are interested in inhibitory signaling, dynamic changes within the ventral CA1 using miniscopes, and also what drugs are we using to target these neuropsychiatric symptoms, and are they actually detrimental for memory? So some foreshadowing on what we're going to get into. So my current graduate student, Sasha, is working to map these brain circuits now that we believe are controlling anxiety-related cognitive decline. And I was really inspired by the Hen Lab. It was the lab next to ours when I was in New York, and they had published a paper just mapping anxiety circuits throughout the brain. And they found the amygdala and hypothalamus were very important for these anxiety-related behaviors. And so I thought, well, let's look at these regions as well and see if they are going awry in Alzheimer's disease and also looking at sex differences. And so we used toxin. So this allows for retrograde labeling. It's not a virus. It acts a little more quickly, so you can actually see these projections in a couple days. So we started here, injected into the 2 regions, and then Sasha also ran some behavior just to confirm what we had seen before. And I don't have any cell count or projection data yet to show you, but we are actively working on this. It turns out CTB is actually very hard to count and label. And so we've seen some projections to ventral CA1, but we actually seen very nice labeling in the entorhinal cortex. And so we're also going to look there, which is another area in Alzheimer's disease that degenerates very early on. And this is kind of what the projections look like into that ventral hippocampal area. Along with the CTB, we are also using a retrograde AAV, using Cree-dependent labeling to actually tag an anxiety experience and then look at those projections as well. So this is ongoing, but we do have some behavior that Sasha has started to look at, and very interesting results from here. So she's actually I looked at that 2 to 6 month range. She is very interested in aging and is going to look at 6, 12, 24 months of age. And so she is still seeing this increased anxiety-like behavior in female mice and males at the 6 month time point. She's looking at an open field test here. So percentage of time in the center, if you're spending less, then we would characterize those mice as more anxious. More time in the center would be less anxious. But what's interesting is that when she ran a test called a marble burying test, where the mice are placed in a cage with 16 marbles and they're allowed to bury for 20 minutes. Burying behavior we call perseverative-like behavior is seen in Alzheimer's patients where they kind of repeat the same motions over and over again with, if their hand or they're going into the same area of a room over and over again. People have related that to marble burying type behavior. And so if the mouse is burying more marbles, they've characterized this as more anxious. And I also found this early on in my studies as well. But when Sasha looked at this 6 month time point, she actually saw decreased marble burying in the AD male and female mice, which is pretty contradictory to what we had seen. So it would technically indicate less anxiety, but Sasha was really tracking these videos and she came to me and said, well, they're just not moving. And so then I started to think, well, is this actually another type of neuropsychiatric symptom as they're getting older that we haven't really looked into before? And so she developed she's called a bout finder. And this is what she developed in her master's program to look at motivation. And she basically takes raw files from ethovision, and mice don't, move continuously. They move kind of in these bouts. If you think of like darting behavior in rats, where they're moving, they have a start and an end time, and then they move again, and then they move again. And so she has about 28 characteristics she can look at here using the raw data from ethovision. And so I'm just going to give you one example of what she's looking at. But in the first 10 minutes, and she's recording for 30 minutes, in the first 10 minutes of this open field, we actually don't see any changes. This is number of bouts. We don't see any differences here. It's really in the last 10 minutes. So if if you're not looking across time, you're not looking at these nuanced behaviors, you're just averaging your data, sometimes you can miss these. And I think that's changing a lot with all of the AI technology that's out there now with behavior recording. And it's just something to always keep in mind. So in these last 10 minutes, this is where we actually see the difference, and we see that females are just not making any bouts here, just not really moving at all. And that's similar to what she had noted in her marble burying task. And so we're starting to think maybe this is a characterization of motivation and apathy. And this has been shown before in a 5X FAD Alzheimer's model, another type of aggressive amyloid model. Again, here, if you're looking at the bars on the right, these are the Alzheimer's mice seeing decrease in marbles buried in both the males and females, kind of matching on to what we're seeing. So that's what we're starting to think this is. So she's looking into this, recording the marble burying data now to also run it through different programs. The other thing, like I mentioned, we're recording in the ventral CA1 area, and so we are looking at activity. This will give us a very dynamic view of what's happening with age and very early on as well. So we're kind of missing those 2 to 4 month early time periods, and so we're hoping to capture that with our miniscope technology. Okay, so I want to move on and talk about, we've characterized this anxiety, we see it in rodents and humans, and now kind of get into how we're treating these age-related mood disorders. And it turns out that we treat these age-related mood disorders the same as we treat any mood disorders. And I think that's something really important to think about, because it may not be beneficial to give the same drugs, obviously, to the aging population that we give to the younger population. And specifically focusing on benzodiazepines. So these are heavily prescribed to treat neuropsychiatric symptoms, anxiety. They're prescribed to millions of adults and especially older adults, and they're 2 times more prescribed in women as well. So a lot of people say they're abused in the younger population, heavily prescribed in the older population. And benzodiazepines are great for a lot of things, especially surgery. They're good at treating anxiety disorders, but the problem is the abuse of benzodiazepines and the prolonged use of benzodiazepines. So the problem is that benzodiazepines have this side effect of anterograde amnesia, or the inability to form new memories. And this has been known since the 80s, since the drug was developed. But no one's really looked at what happens if the older population are taking these drugs for a long period of time. And so recently, people have started looking at the memory deficits associated with benzodiazepines and also looking at the brain regions that are affected. So our question became, do benzodiazepines increase the risk of dementia? We're working with a collaborator here, Dr. Joanna Dabrowska, in pharmacology, and my other graduate student is very interested in this project as well. And if you look in the human literature, it's highly controversial on whether benzodiazepines increase your risk of dementia, with about half saying yes and about half saying no. And so we turned to the rodent literature, and many studies have shown, impaired working and spatial memory in mice. And more recently, people have started looking at what happens in aged mice and prolonged use. So this is a study looking at prolonged use in middle-aged mice, showing memory deficits and also changes in the number of spines in the hippocampus. So also changing the brain as well. And it's still unclear whether the brain can bounce back if you stop taking this medication. So still a lot of unanswered questions on these drugs that are used to treat these neuropsychiatric symptoms. So we've also started working on a review to better understand whether benzodiazepines are associated with dementia. And it turns out that it's actually pretty complicated. So we found 9 studies that had no association. We found 9 studies that had a strong association, and then a few in between. And we're working on this now, and it's basically just a bit messy with controlling for different comorbidities. Controlling for sex may not be the best thing, whether you're starting with different baselines, cognitively normal versus if you're starting with mild cognitively impaired patients, longitudinal versus cross-sectional. So a lot of things to think about. We're working on this now, but still very complicated. So we wanted to at least try to understand the mechanisms behind these benzodiazepines. So what brain regions, what neuronal mechanisms are responsible for this memory impairment that many people have seen? How are the neuronal ensembles altered after alprazolam injection? So using our activity-dependent tagging mouse line for this, and then also looking at whether males and females respond differently to the drug. So we're using a similar setup that we had in my first study, where we're tagging those cells after either an injection of saline or alprazolam. So alprazolam is Xanax. It's the most common benzodiazepine prescribed, so we are using that. We inject that 30 minutes before their training. We place them in the chamber, shock 3 times in a context, tag those cells in green. We dark house the mice for a few days to prevent any non-specific binding. And then we re-expose them 5 days later, and we look at freezing behavior as a proxy for memory. And then 1 hour after that, we again look at that CFOS, that immediate early gene, and then we can look at the overlap. And so this study is also recently published. Feel free to scan the QR code if you want more details. I'm also happy to answer questions on all of these at the end. But what we found was after a single injection of alprazolam, there were memory deficits in both males and females. This is in C here. You'll also notice that during training, there is a lot of freezing in the benzodiazepine-treated mice. That's because the benzodiazepines are a slight sedative as well. But we wanted to make sure that mice were still experiencing the shock, or they're still learning that this is an aversive chamber. And so we did a shock reactivity test where we just kept increasing the intensity of the shock in all groups, and we found that around 0.2, 0.3, all mice were responding. So they are experiencing the shock in a similar way. And this video is just basically showing the re-exposure of a saline mouse, what it looks like freezing versus an alprazolam-treated mouse. So we saw the memory impairment that others had shown. Then we wanted to look at the neuronal ensembles. So we looked first in the dorsal hippocampus. we saw no differences, no changes in males. So I'm only showing the female data, but I will get into maybe why we haven't seen those differences in males. So only differences in females, but also not what we had hypothesized. So we actually saw an increase in encoding cells with alprazolam in the dorsal CA1, and we also saw an increase in engram cells in the dorsal CA3. And we had hypothesized that we would kind of see a dampening of everything. So benzodiazepines activate, GABA and inhibitory signaling. So we thought, well, this is very strange. But the strength of an engram doesn't necessarily mean the strength of a memory. So we looked in the ventral hippocampus. Here is where we actually saw the decrease. Again, we looked in ventral in males. We see no differences. So again, I'm only showing females here. We see a decrease in that FOS activation, specifically in the ventral CA1 of female mice. So again, similar patterns of these brain regions that we've seen before, right? The ventral CA1, the dorsal CA3 in females seem to be quite vulnerable. And so we have a bit of a hypothesis on how this is occurring because we do see the increase in encoding in engram, but decrease in retrieval. And so excitation inhibition balance is really essential for memory formation. And we think this is disrupted with alprazolam. And we're specifically interested in somatostatin inhibitory interneurons because they modulate the excitability of the dorsal CA1, and they are controlled by some downstream regions. And so what we're hypothesizing is that alprazolam may alter the circuit so that you would have a blunting basically of the somatostatin neurons here, and you would get an increase in excitability in the dorsal CA1, and that's kind of what we're seeing in the females. And that would still result in a memory impairment because the excitation inhibition balance is not where it needs to be. So Cameron, my graduate student, thought, well, let's sacrifice the mice 1 hour after an encoding experience and then look at somatostatin activated cells. So any of the somatostatin cells that are overlapped with FOS and see if those differ. And again, ran males and females. And this is actually where we finally see the difference in males now. We're seeing a decrease in these SST inhibitory neurons in the alprazolam treated mice in both males and females. Also seeing a baseline sex difference there as well. And so from here, we really want to understand maybe this is a potential mechanism that benzodiazepines are working through to impair memory. And also, is this something that would impair memory if you keep taking these drugs for long periods of time? And so from here, we are using our new SST Cree mice to manipulate these circuits while benzodiazepines are on board to actually try and rescue the memory to determine if this is a true circuit that alprazolam is working through. So to summarize all of that, we saw memory impairments in both males and females after a single injection, but we only see changes in female mice, specifically increases in activation in the dorsal hippocampus, decreases in retrieval in the ventral hippocampus. Now, the reason that males and females may differ in our initial analysis is because we have only really looked at the hippocampal regions. We may see differences in males in different brain regions. We know that at baseline, males and females utilize different regions during fear memory retrieval. So right now we are looking into counting different brain regions. We also know that alprazolam is lipid soluble, so it may be metabolized more slowly or build up in the fat tissue in females. And we have only looked at one dose here, so dosing could matter as well. So from here, we are also, again, interested in that translational science, is what we're seeing what we would see in a human cohort. And so we have partnered with Dr. Namhee Kim, who's a biostatistician here, to look at the Rush Memory and Aging Project. So Rush is right down the road in Chicago, and they have a large, awesome human data set, and we are going to look at the impact of anxiolytic medication on AD progression, looking at sex differences and also underrepresented minorities. And so just to give you a little glimpse into this, we're still working on this, it's in prep, but we have started to see a similar pattern where what I've highlighted here. So the first highlighted box is just anxiety diagnosis. It is related to a decrease in global cognition. But what's more interesting is that the second highlighted box is the anxiety medication. And this has a greater effect size alone than with just looking at anxiety, also highly significant related to global cognition, a decrease in global cognition. And same thing for global AD pathology. It's really the medication that's driving this, and something that maybe was driving in our previous study that we had shown as well. So it's not just the anxiety, it's maybe the drugs that are used to treat the anxiety as well. Okay, so because we're still, ongoing studies with that, but we're thinking that benzodiazepines are probably not very beneficial to give to an aged population that are already vulnerable to memory impairments. And so we started thinking about non-traditional therapeutics to treat neuropsychiatric symptoms and Alzheimer's disease. And so we've partnered with Beth Stutzman, who's the chair of my center, and Shiwa, who is a medicinal chemist. And we are looking at using psychedelics. So psychedelics have long been used in the mental health space, but have not really been looked at for treating memory disorders. And when I was reading about psychedelics, I thought, well, they're fixing everything that's wrong in Alzheimer's too. So why don't we at least try? And now am I saying, should we give psychedelics to Alzheimer's patients? Not necessarily. But if we can use this as a treatment earlier on, or even harness some of the aspects of psychedelics with our medicinal chemist and take out all of the good parts and make those into new medications, that's what we're really interested in understanding. And so we decided to use psilocybin. It's handled very well. It received FDA approval in 2019 for major depressive disorder and also enhances neuroplasticity, reduces neuroinflammation, is good for cognitive flexibility, and this drug targets the serotonergic system. And so brain-wide networks are often altered with psilocybin. More recently, psilocybin has been shown to extend lifespan, improve survival of aged mice, induces rapid and persistent growth of dendritic spines, all these beneficial effects, and looking at plasticity through different mechanisms. And so we decided to test this in our APPPS1 line. And we injected saline or psilocybin, 3 mg per kg, which is a moderate dose, again in that same 6 to 8 month time point. And we injected once, we waited 7 days, and then we tested behavior. And then some of these brains were given to the Beth Stutzman lab for electrophysiology studies. So what we saw after giving psilocybin, again, this is 7 days later, this is looking at a zero maze. So more time spent in the closed arms represents anxiety, more open arm time, less anxiety. But what we actually saw with psilocybin was some hints at exploratory behavior being increased. And this has been shown by others as well. And in female mice, we specifically see increase in distance traveled in the psilocybin groups in both the control and AD. However, for anxiety-like behaviors, we don't really see any effects in females, but we see an increase in open arm time in the control psilocybin-treated males. And also we see time highly mobile again, and we're kind of terming this exploratory behavior in the females in both the psilocybin-treated groups. And then we looked at latency to enter the closed arm. So they start in the open area, and we look at the latency for the mouse to enter the closed area. Again, a longer period here in the control males treated with psilocybin, which matches their time in the open arms as well. This is still a preliminary study. We don't have a ton of N here for every group, but it is an interesting trend that we're seeing. In terms of slice electrophysiology, this is looking at an input-output curve. And what that means is there's a stimulating electrode, we increase that over time, and we look at the excitatory potentials. And I had mentioned this before, but this idea of hyperexcitability. So in this mouse model, there has been hyperexcitability seen, especially in electrophysiology studies. And so in the AD saline, that's this top row here, this light blue, we're seeing that increased hyperexcitability, but the psilocybin treated AD mice, we see a reduction, significant reduction here back to control levels, which is really exciting. We're combining males and females here because we just don't have the N, but we're really excited to look at those sex differences as well. So from here, we went a step further. We looked at whole cell patch clamp at the neuron itself. And you can see that hyperexcitability. This is calcium evoked release. It's very high in the AD saline. When you treat with psilocybin, completely significantly reduces that. And this is also seen in the bar graph. We're not sure exactly the mechanism through which this is happening, but it may have something again to do with that excitation inhibition balance. So we're seeing a lot more inhibitory potentials in the psilocybin treated group compared to the saline treated group. So again, it may be balancing that excitation inhibition. And we also looked to see, again, if this is translatable using human-induced neurons derived from iPSCs from AD patients, again, from that ROS-MAP study. And we see a similar pattern. So the top row here, I know it's a ton of data, but it's all essentially the same pattern. The top row is short-term dose. The bottom is long-term dose. That means overnight. Short-term is 1 hour. With psilocybin increasing concentrations, you see a decrease in that hyperexcitability or that caffeine evoked release, also in neurons from human patients. So very exciting. And so we decided to look at psilocybin with our mini scopes. And so this is a video. We injected psilocybin and we just started to record right away. We just wanted to see what this would look like in real time. And we continued recording across several weeks, and we see cell amplitude, cell strength really staying increased even 4 weeks after psilocybin injection. And we've actually gone longer than this now, and we're seeing this up to about 12 weeks. So the brain has changed for that long, maybe longer. Let me get to the where it gets really. So it's about like a minute into psilocybin injection. This is where you really start to see a lot of the firing. Kind of looks like a Christmas tree lighting up, but it's one of my favorite videos to show. So we also looked after miniscope recording to look at correlations of the cells firing. And at baseline, not highly correlated. After psilocybin, that's where we see a lot more cells come online and also seem to be more highly correlated with each other, both a positive and negative, very balanced correlation. This is just one example. This is just an example in an aged mouse, not an Alzheimer's mouse, but that is the direction that we are going. So we think maybe psilocybin could be a novel therapeutic for AD. Obviously, further study is required. We see some changes in anxiety-like behaviors. I didn't show the fear conditioning. We're still adding to those groups. There is a trend towards increased performance in males only, but not in females. So I think the dosing will have to matter. But we have seen a lot of changes within the hippocampal excitability, both all the way from slice to human-induced neurons and into our rodent in vivo imaging as well. So we're very excited about that. And the last thing I'm going to talk about that I'll wrap up with and leave enough time for questions is this idea of what happens if you're stressed on top of all of this. And when I had first started this study, everyone kept asking me, well, what happens? Because, these animals are not stressed. They're just showing anxiety because they have these Alzheimer's pathology. And what happens if you stress these animals? And we hadn't done that experiment. I was holding off. And then once I started my own lab, we started looking into this, and it was really spurred by one of our collaborators. So we are working with Dr. Nicole Ferrara, who she is actually in the stress center here, and she looks a lot at adolescent stress, isolation, instability, does amazing work there. And my lab tech, Lainey, actually went to one of her talks and was so inspired, and after said, well, what about isolation and aging in Alzheimer's? And after we experience something stressful, do we respond differently if we have friends versus being alone? And it really started us thinking about the COVID-19 pandemic, and a lot of elderly patients were isolated. So and a lot of the isolation field has been focused on adolescence and development, and not a lot looking at, well, what happens, and stressing early on and then looking across, but what happens if you just stress an already aged or AD mouse, and then either put them back into group housing or keep them separated and they experience something stressful, what happens? And so we are looking at social isolation's impact on memory and cognitive decline. We're looking at sociability, which is a very new area for me, and Nicole is helping me with that. And also, again, looking at the brain regions and circuits that are impacted by this. So really looking at how social isolation or whether social isolation can drive Alzheimer's pathology. So socialization is really important for survival. We thrive being social creatures, and mice also thrive being social creatures. And it's very important. And actually, until I had really looked kind of into this field, one of the protectors against dementia transition, one of the biggest ones is sociability. And so there's a need to understand why this is. And a recent review had published that reducing AD prevalence by 4% would be to eliminate social isolation as a risk factor, and it would provide a greater reduction than, combating physical activity, which I'm also a big proponent for, and hypertension. So this is an area that we've become pretty interested in understanding. We know that loneliness and isolation is bad. We know that negatively affects health. It can increase risky behaviors in the elderly, can affect mental health, can decrease cognition, and you're 50% more likely to develop dementia as well. In the rodent literature, like I said, the mechanisms haven't really been looked at in the age mice or AD mice. A study in middle-aged mice, again, 30 weeks of isolation is a long time. We kind of want to make a more translational study where we're isolating them, maybe putting them back into group house. And so, nevertheless, with isolation, you see anxiety behavior, memory impairment, changes in the hippocampus as well. And so we started with a very simple timeline, looking at, again, that 4 to 6 month time point, males and females, getting baseline behaviors of open field and social interaction, and then isolating them, group housing, putting them through something stressful like a fear conditioning paradigm. That way we could kind of look at memory, but also look at what happens after isolation and a stressor in both anxiety and social interaction. So the most that they're isolated is about 8 days, so pretty acute. And so what we found was very striking initially. All of these graphs are going to be set up the same way. So post, which would be that day 8 minus pre, which would be the start, looking at control group house, socially isolated, AD group house, socially isolated, female and male. It's a lot going on, I know, but we've tried to group it into something that is easily understandable. So we looked at the difference in center time. Again, center time would be less anxious, perimeter would be a bit more anxious. We're seeing that after stress, there's an increase in anxiety-like behavior, specifically in the female mice, an increase in center time actually in male. So they seem to be unaffected. And we're actually seeing the greatest difference in the group housed mice. So that means we're thinking the stressor is more of a factor here than the isolation, which has very little change. But again, this timeline, it's very hard to separate out the stressor from the isolation, and that's something that we're going to do in the future. We then looked at social interaction. And so here's an example of mice interacting. This would be a nose-nose, a little bit of a nose body there, and then nose anal genital you'll see in a second. And then in the second video, you'll notice that mouse is not engaging. So these are all of the different types of behavior you could see. The novel mouse is marked in red here. The test mouse, that's the behavior that we're measuring, how they interact with the novel partner here. And what we saw was a sex effect in a lot of the social behaviors, but more specifically, an increase in huddling behavior, again, after stress and isolation in all the female mice, control and AD. Not much is changing in the males in sociability before and after stress or before and after isolation. So huddling behavior is something that we're looking into. This is a very new field for us. So if anyone has ideas on why this is happening, I would love to hear more about it. For a long time, people thought huddling was for temperature, but maybe here we're thinking of this more as like a compensatory mechanism to stress. So lastly, when we looked at the fear retention memory, we saw some very different results than again going against what we thought. Um, so in again, that freezing behavior is a proxy for memory here. We don't see any differences in fear retention in control mice, whether they were group housed or isolated. So memory is intact. Again, this isolation is acute. We still see the memory deficit in 80 mice that are group housed. That's typically what we've always seen. But in Alzheimer's mice that were socially isolated, we actually see enhanced fear expression. And we're not really sure why this is or what's happening, but we're thinking that maybe this is some type of PTSD-like phenomenon. So they're isolated, which is enhancing the fear retrieve. It's kind of overriding the memory impairment, preventing extinction. So this has all been shown before in the PTSD literature, but we did not expect this. Again, if anyone has ideas, let me know. In males, we see that typical pattern that's always been published on. So social isolation decreased memory here in the controls and further decreased it in the AD mice as well. So from here, we are also using those immediate early genes. This time we wanted to look at activated parvalbumin inhibitory interneurons. We know that from the early life stress literature, there's this decrease in inhibition excitation balance. Again, same theme that we've been on through the talk. We're really interested in parvalbumin here because we know that these neurons also respond to sex hormones differently. And so we really wanted to understand the activation of PV. This is very preliminary. We only have counts for females right now, but I love to show new data. So it's a hint. Come back in a few months for more maybe, but whenever we can get this paper out. But we are seeing an increase in PV activation, specifically in the ventral CA1 in female mice, in female AD mice specifically. So this could account for why we're seeing those memory deficits in AD mice, just overactivation of inhibitory cells there. So let me summarize this and then I'll talk a little bit about future directions and then open it up for questions. So we see increased anxiety after stress in female mice, increased huddling behavior, decreases in social interactions in females. Interestingly, increased fear retention in the female AD mice, but decreased fear memory in socially isolated males. And we are going to continue our cell activation analysis and also add an aged component as well. So our future directions, we are heading into menopause. So we received an internal grant from RFU. I've been wanting to get into this area for a long time. And so we received a grant to collect some preliminary data and then are applying for an R01. So we are looking at whether menopause or how menopause accelerates AD pathology in women. A lot of people before me have shown changes in rodents, in humans, structural changes, metabolic changes. And then we really wanted to model this in the best way in rodents. So we're using the VCD model to study menopause transition. So this is a chemically induced menopause. And this results in the slow loss of ovarian follicles across time. So we can look at anxiety, cognition, sleep across these transition periods, specifically looking at perimenopause, looking at brain changes. Oops. And also looking at cell activity across these transitions as well using our miniscope technology. We have confirmed that this model does work in our hands. So we've shown earlier anxiety-like behavior. We've shown weight gain with the menopause, and we've also shown irregularity in cycling. And in this study, we are going to be using a different Alzheimer's model, the APP-NLGF, which is a knock-in model. So it's a little bit more translational and allows us to look more at progression rather than that accelerated pathology. We're also seeing cognitive decline post-menopause in these mice, and we think it might rebound. So if we wait about 100 days later, we are seeing actually intact memory again. It's something that we're exploring. But again, all preliminary data, we don't have any controls yet, just looking at VCD in AD mice here. And again, using our miniscope, so we're going into a new brain region. We've been in the ventral CA1, troubleshooting that one for a while. Now we're moving into the dorsal CA3 here for our menopause project because that's where we see a lot of the changes in females. And so we have successfully implanted it here now too, so it's exciting. So hopefully, I know that it's a lot, the Alzheimer's exposome, but I wanted to touch on kind of all of the projects that we're doing and how they relate to each other and how important it is to look at this multifaceted disease from a lot of different lenses while also always taking into consideration sex. And we are limited to sex as a binary in our studies here. So we don't have the chance to really look at gender, but that is very, very important in the human literature as well. And I hopefully I've convinced you that there are tons of individual differences across the lifespan that can contribute to neurodegeneration, and we are trying to uncover all of these slowly. So with that, I want to thank my lab. I don't do this alone. You'll notice on every slide I have a collaborator. I love team science. If you want any protocols for anything, some are on my website. If that's outdated, just shoot me an email if you want to know about something. But yeah, shout out to the lab. We grow in the summer with all of our undergrad volunteers, and then we shrink back down in the fall. Cameron and Sasha are my two graduate students. Lainey is my lab tech. She helped me set up the lab, has been with me through it all of our funding sources and all of our collaborators, the people that are highlighted here are the ones who did a majority of work in this talk. Feel free to follow me and social media, scan the QR code for more info. And with that, I thank you and I'm happy to take any questions.
Well, thank you very much, Holly. I know it's always difficult in these virtual, but hopefully you can see some of the props. It was quite a whirlwind, very
Yeah.
very. So I had lots of questions, but we only have 4 more minutes. So I think there might be a question from the audience too, but I will please. I better take a look at you before I say this. But if you have one, please raise your hand. And I see Bonnie has one. And then I want to talk about benzodiazepines just for a moment. Bonnie, go ahead.
Hi, Dr. Hunsberger. Really interesting talk. Thank you so much. I'm just really curious about the activation differences in the hippocampus you saw between males and females. Did you look within the hippocampus if there were differences? And then my second question is if you looked at the retrosplenial cortex at all and how that might contribute to the fear circuitry.
We haven't looked at retrosplenial, but that's a great area. We have not looked there yet, specifically at the engrams. I think it is in our brain-wide network map. I'm not sure how correlated. I can't remember off the top of my head, but it's in that paper. But we have not looked at the activation in that area yet. But we did look at the hippocampal activation, and that is different in males and females.
What struck me too is that it goes in opposite directions if I'm
Yeah,
Mhm.
yeah, yeah.
just really and that's just another plug for why just because you see a certain direction in males doesn't mean you're going to see the same direct. And a couple times you even said, oh, this is like, we weren't expecting this.
Right, right.
But I think that's a really good, important lesson for people that,
Mhm.
yeah, it looks like it's can be quite different.
Mhm. Yeah, thank
Anything
you.
else, Bonnie? No?
It's okay. I
Okay.
will email you if I have further questions.
Yeah, yeah, please, please.
Um, So the benzodiazepines is really interesting. I also just self-disclosure, my mom was prescribed them when she had some dementia. I was like, I don't think this is the right thing for you. And so because she actually said, and she actually disclosed to me that she felt more agitated
Mhm.
taking them, even though it's supposed to exactly do the opposite. But it reminds me a little bit of Ritalin, right? Like there are different just because a drug does one thing in one age doesn't mean it's going to do the same have the same kind of output at a different age. And so it's not like radical thinking, but for
Right.
some reason that is really prescribed, highly prescribed as you demonstrated. And in your mice, I think I might be mistaken, but did you also see higher levels of activity in the females only, I think. Do you remember that?
In
Because you had a lot of different data
yeah, yeah, in the active brain activation or activity and behavior.
in behavior. Yeah, just talking about it behavior right now.
So we see decreased freezing in both sexes with alprazolam.
But that was with the memory, right?
Yeah,
Maybe I'm thinking about the psilocybin. I think
yeah,
maybe
psilocybin more active. Yes.
that's also so
Yeah.
interesting. And I will just say thank you so much because we're out of time, I
Oh
guess.
no.
But yeah, thank you, fantastic, Rowan. You gave us a lot to think about. I wanted to just bring up with your benzodiazepines and thinking about review article you're doing where you see positive, negative, and no change, I know you're on the right track because you talked about like, hey, maybe sex is a covariate isn't the right way
Mhm.
to do it. But of course, I immediately thought of, hey, sex, age, like I mean, age might also different, or SES status or ethnicity, all of that probably plays a role, as well as your healthy self bias, which you touched on too. So we look forward to that and many more publications coming out of your
Thank
lab. Thank you
you.
so much for your time.
Thank you. Thank you all for your attention.
Bye,
Thank
everyone.
you.